Hello, everybody, and welcome back to BioSC 140, Human Physiology. This is the part 2 of the communication lecture video. So, Claude Bernard said the constancy of the internal environment is the condition for free and independent life. There are a lot of different chemical reactions that take place within your body, within human bodies, that allow that human body to continue to live. Those chemical reactions that allow life to happen require very specific environments, very specific conditions in order for them to happen. Walter B. Cannon coined the word homeostasis. It means same within oneself. And he had 4 postulates about homeostasis. And I want you to know all 4 of these postulates, and I want you to know examples of each of them. So the 4 postulates were: the autonomic nervous system plays an important role in preserving the fitness of the internal environment. By fitness, it's the stability, by maintaining the ability of that internal environment to have the conditions for those chemical reactions to persist. Number 2, systems are under tonic control. We'll get into what that means. We have slides for each one of these points coming up. Antagonistic controls are important. You should know a few things about antagonistic controls from week 1 lab. And chemicals can have different effects on different tissues. All right, moving right ahead. The autonomic nervous system plays an important role. So you should already know a few ways that the autonomic nervous system plays an important role in maintaining homeostasis. We've already talked about in lab how the autonomic nervous system can affect our heart rate. It can affect our body temperature through sweat production. So there's 2 main branches of the autonomic nervous system: the parasympathetic and the sympathetic branches. There's the parasympathetic. People describe it as the rest and digest branch. The parasympathetic is more active when you are resting and digesting, and it promotes functions, it promotes bodily activities that your body does, that your body needs to do when resting and digesting. The sympathetic branch is the fight or flight branch. And when its activity increases, it prepares you to fight or flight. And by flight, it means run. So if you're a human 100,000 years ago in the woods and you come across a bear, you need to fight that bear or you need to run away from that bear. That's when the sympathetic nervous system is going to increase its activity, and the sympathetic nervous system promotes those functions that would help you with either fighting a bear or running away from a bear. Both of these branches work below the level of consciousness. Now, this picture to the side, you have seen it before, and you'll see it again during our nervous system lectures. You don't need to memorize every single one on this picture. Read over them and get a good idea of what they are. The ones that we specifically talk about are the ones you need to memorize. So sweat, heart rate, ones that we specifically talk about. Tonic control involves modulated activity of controller. So tonic control involves a modulated activity of a controller. So what is tonic control? Tonic control means there's always some level of signal. Tonic control means there's always some level of signal within your nervous system and in your endocrine system. So what does that mean? Well, let's say you're resting and digesting. Your sympathetic nervous system never completely turns off. there's always some rate, some level of signal happening in your sympathetic nervous system. Now, if you remember, action potentials or signals in your nervous system always have the same amplitude. Every signal has the same amplitude, has the same force, the same impact. You can't send a stronger signal or a weaker signal when you send an action potential down the axon of a nerve. It's the same amplitude every time. That's why all these hash marks right here, same amplitude. What you can modify is the frequency. You have a low frequency within your sympathetic nervous system. When you are resting and digesting, you have a high frequency within your parasympathetic nervous system. They never completely turn off. When you are fighting that bear or running away from that bear, don't fight bears, run away. You have a high frequency in your sympathetic nervous system and a low frequency in your parasympathetic nervous system. You are never able to completely turn that frequency down to zero. So increase or decrease activity from a single controller increases or decreases a parameter. Within the endocrine system, you see tonic control also. You always have some, for all of the hormones, there's always some level of that hormone within your body. You never completely get rid of a hormone. There's always some level of it, and that level can change. Tonic control. So men and women have the same hormones. This hormone right here, HGC, sorry, it should be HCG, sorry, that's a typo, it should be HCG, human chorionic gonadotropin. Pregnancy tests, at-home pregnancy tests, they test for the presence of HCG. Women always have some level of HCG in their bodies. When they get pregnant, that level increases a lot, and it's that increase that these home pregnancy tests test for. Women who are not pregnant still have some level. There's still tonic control, but it increases greatly when they're pregnant. Men and women actually have the same hormones. So men have some level of HCG in their bodies at all time also. The way that, here's a fun little mnemonic to remember HCG as a pregnancy test hormone. HCG, holy cow, girl, you're pregnant. All right. So sweat glands are exclusively under sympathetic control. There's tonic control, so there's always some level of, some frequency of signals being sent to them. And there's no parasympathetic innervation of your sweat glands. Now, we've gone over this in lab. It's sympathetic nervous system only. When you're at rest, your sympathetic nervous system has a low level, a low frequency of signals being sent. You have a low sweat rate, a low level of sympathetic activity. When you are shocked, when you have to fight or flight, you have an increased level of sympathetic activity, an increased frequency of signals being sent in your sympathetic nervous system. This will increase your sweat rate. Increased levels of sympathetic activity increases your sweat rate. Now let's think about what we know about heart rate. So heart rate, you have a dual innervation. Parasympathetic decreases heart rate. Sympathetic nervous system increases heart rate. At rest, you have low sympathetic, so low stimulation to increase heart rate. You have high parasympathetic, so high stimulation for decreasing heart rate. You have a low heart rate. When you're shocked, when you're excited, you have a high level of sympathetic nervous system activity, increasing your heart rate. and a low level of parasympathetic activity decreasing your heart rate. Two controllers oppose each other to provide antagonistic control. So this gets into the next postulate, and that's the one of antagonistic control. With our heart, we have antagonistic control. We have a pair of regulators that oppose each other's action. Increased sympathetic activity, increased heart rate. Decreased sympathetic activity, decreased heart rate. With parasympathetic, it's increase parasympathetic activity, you decrease heart rate. You decrease parasympathetic activity, you increase heart rate. Sympathetic nervous system, direct correlation. Parasympathetic is inverse. With antagonistic controls, so if you remember back to lab quiz 1, we talked about how the water bath was similar and not similar to the human homeostatic situation. And we said that it's not a good example of human homeostasis because it only has unidirectional control, one direction control. Antagonistic control requires two directions, control in both directions. And things that have dual sympathetic and parasympathetic innervation, usually, not always, but usually there's actually one example where sympathetic activity and parasympathetic activity both promote the action. We'll get to that in the nervous system lecture. But most of the time there's dual innervation where the sympathetic and parasympathetic branches are innervating something. They have opposite effects. They provide antagonistic control. Hormones can also provide antagonistic control. Now we've gone over these two hormones already, glucagon and insulin. If your blood sugar levels go up, you produce more insulin, you produce less glucagon. If blood sugar levels go down, you produce more glucagon, you produce less insulin. You never stop producing insulin, you never stop producing glucagon. There's always some level of each, but that ratio can change. And these two hormones work antagonistically with regards to blood glucose levels. Chemicals can have different effects in different tissues. So a receptor isoform are different forms of a same receptor. They're receptors that react to the same signal, but they cause different things to happen. So receptor isoforms are receptors that respond to the same signal, but they cause different things to happen. They have different outputs. So let's look at this alpha receptor and this beta 2 receptor. So in the intestines, we have alpha receptors on our intestinal blood vessels. When epinephrine reacts with that receptor, it causes vasoconstriction. So the signal is epinephrine, the response is vasoconstriction. When epinephrine reacts with our beta 2 receptor and on the blood vessel of our skeletal muscle, it causes the vessel to dilate. So same signal, different output. Different chemicals can have different effects on different tissues. Receptor isoform, same signal, different output. Homeostatic control systems maintain parameters near a set point. So local control involves a response from the region where local control involves a response from the region where the change occurred. So with local control, we usually have, it's called intrinsic control or auto-regulation. Local control is when the local area controls its own regulation. Control comes from within the system. So right here, These cells around the blood vessel, around the blood vessel, notice that there's low oxygen levels. These cells are going to send signals to the neighboring blood vessel. It should dilate and increase blood flow, which is going to increase oxygen levels. Local control typically involves autocrine or paracrine signals. Extrinsic control is when control comes from outside the system, when control comes from somewhere else. So in extrinsic control, an integrating center is removed from the affected area. So there's three main types. We have neural. So with extrinsic control, this is a processing center away from the tissue or away from that organ controlling it. So with the nervous system, we have the central nervous system doing all the processing, with the central nervous system deciding what action should be taken. The central nervous system is outside the heart, for example. That's extrinsic control. So the autonomic nervous system telling the heart to beat faster or slower is extrinsic control. Humoral control is your endocrine system. It's hormones. This is epinephrine, a neurohormone, being released into your bloodstream and having an effect on your heart rate. Hormones, type of extrinsic control. You can also have behavioral control of some of your variables. So body temperature is homeostatically regulated. You can help your body control that variable by how you behave. So if you're cold, you can put clothes on. You can go for a run. If you're hot, you can go move into the shade. Behavior is a way to control some of those homeostatically controlled variables. You could even say that behavior can have an effect on blood pressure. You can do meditation, help have an effect on it. So set points are variable. Set points are variable. Remember, a set point is the target value for a homeostatically controlled variable. It's the ideal value for that variable at that time, at that specific time. That ideal level might change in different circumstances. So when you get a fever, your body, the set point for temperature rises when you get a fever. Body temperature actually fluctuates throughout the day. You can see at night your body temperature drops a little bit. During the day it rises some. Your body temperature, female body temperature actually drops during ovulation also. So for a certain period of time throughout their cycle, body temperature will actually drop also. So set points are variable. Feed-forward control allows the body to anticipate change. So your body can anticipate the change of you waking up. Body temperature regulated is in part by hormones, by cortisol. Your body will increase cortisol production shortly before you wake up, so that when you wake up, your body temperature starts to rise. Cortisol production is influenced by your circadian rhythm. Morning people, so your body temperature goes up before you awaken. That circadian rhythm, that cortisol production ramps up before you wake up. So you're good to go when you wake up. You're a morning person. Feed-forward control is also a little better visualized, a little better experience with food. So me just seeing Zachary's pizza, seeing this photo right here, I start to salivate a little bit. Salivation is an anticipatory reflex. An anticipatory reflex. It anticipates a change. It's feed-forward control. Feed-forward control was famously examined by Ivan Pavlov. Many people have probably heard about Pavlov's dogs. So Pavlov, he experimented in this area with dogs. So you see this contraption, this thing right here coming off the dog. That thing is a saliva catching container for this dog. And what Pavlov did was every time he fed his dogs, he rang a bell before doing so. So he would ring a bell and then give his dogs food. And eventually what he found was that he could just ring his bell and his dog would start to salivate. They didn't need to see the food. He'd just ring the bell and they would anticipate, they'd have feed-forward control on their salivatory glands. They'd start to salivate. Reflexes involve extrinsic control. So this next part, there's something that I want to bring up first, as students often get this mixed up. We're going to cover the 5 characteristics of reflexes, and then we're going to cover the 5 parts of a reflex arc. So we have 5 characteristics. We have the 5 parts of a reflex arc. 5 parts of a reflex arc, 5 characteristics. Keep them separate in your minds. So the characteristics, involuntary. Reflexes are involuntary, so there's no conscious control. With regard to the baroreceptor reflex, which we'll talk a lot about when we talk about the cardiovascular system, you can't think really hard and increase your blood pressure. You can't think body, increase your blood pressure and do it. No conscious control. When the doctor hits your knee with his reflex hammer, there's no conscious control to you moving your leg forward. It's a reflex. There needs to be adequate stimulus. You need to exceed a threshold. So when the doctor hits your knee with his reflex hammer, he doesn't use a reflex feather. You know, if touching your knee with a feather is not an adequate stimulus to get the response. You need adequate stimulus. You need to use that hammer. All reflexes have a specific receptor that responds to a specific stimulus. So with the knee-jerk reflex, it's a stretch receptor. The doctor hits your patellar tendon with the hammer, it stretches the tendon, and your body, and there's a stretch receptor that responds to it, that initiates the reflex. Patterned, or it's stereotyped, means you get the same result every time. When the doctor hits your knee with the hammer, your patellar tendon, you don't contract your hamstrings, you contract your quadriceps. It's the same every time. It's graded. The size of the response determines the size of the stimulus determines the size of the response. The size of the stimulus determines the size of the response. So if the doctor hits your patellar tendon harder, you'll get a bigger response. All reflexes follow an arc. So all reflexes follow this pathway. There needs to be some sort of stimulus that a receptor detects. So part 1 of the arc is the receptor. Remember, the receptors detect, there's a specific receptor that detects a specific response, the receptor. That receptor needs to send a signal via an afferent sensory fiber. That signal needs to be integrated by the central nervous system. That central nervous system needs to decide what to do with that information, integration of information. And once the central nervous system has decided what to do, there is a motor output via an efferent fiber. Motor output via efferent fiber. And that efferent fiber is going to send a signal to an effector, or something that can cause a response, something that can cause a change, something that can cause a response. Reflexes can be simple but important somatic nerve pathways. This is an example of the knee-jerk reflex, which we'll see in lab. We have our stretch receptor, our muscle spindle, our stretch receptor. We have our nerve endings coming in. So afferent sensory nerve going to the central nervous system, the integrator, sending signals via the effector, the efferent nervous system, to the thing that can affect change, the effector, in this case, muscles. If you have any questions, please send me a message and let me know. Have a great day.